All right. My name is May Daher. I'm an assistant professor at MD Anderson in the Department of Stem Cell Transplant and Cellular Therapy. And I would like to thank the organizers for the invitation. And it's my pleasure to be here today to talk to you about CRISPR optimized cellular therapies. So these are my disclosures. And I won't spend too much time on the background since the audience is very familiar with CRISPR and what it does, basically. But we all know that CRISPR has revolutionized the field of molecular biology and has won the Chemistry Nobel Prize in 2020. And if we think about it, really, it was developed just 9 years ago, and the first results were published in Science in 2012. And since then, there has been an explosion of use of CRISPR in multiple fields, including biology, chemistry, medicine, agriculture, etc. So the scalability of this approach is amazing, and it's due to the elegance, the simplicity of the approach, and its reproducibility. And it really relies on these two simple components, the Cas9 protein, which acts as a scissors to cut the DNA in a double-stranded fashion, and the guide RNA, which is a small piece of RNA that binds to the Cas9 protein and guides it to a specific site of the DNA to be specifically cut. So from a clinical standpoint, really, there has been also an explosion of clinical trials recently testing ex vivo and in vivo CRISPR gene editing strategies to treat multiple diseases, including refractory cancers, including sickle cell beta thalassemia. You've heard about immunodeficiency syndromes as well, and more recently, even amyloidosis. And if you go to clinicaltrials.gov and search for CRISPR gene editing cell therapies, you can find over 15 clinical trials describing these approaches. Most of them are ex vivo clinical trials. Most of them are phase 1 and testing this approach in CAR T cells or hematopoietic stem cells. So at MD Anderson, we're very interested in CRISPR technology as well to enhance the function of our adoptive cellular therapies, and we are mostly focusing on virus-specific T cells and NK cells and CAR-NK cells, so two different platforms, and I'll give you examples of both. So I'll start with the CRISPR-modified virus-specific T cells. We all know that viral infections remain a leading cause of morbidity and mortality after stem cell transplantation and in patients who are immunosuppressed with cancer. Prophylactic therapies such as letermovir or ganciclovir are effective in reducing somewhat the risk of these viral infections, but however, breakthrough infections still occur. And the available pharmacotherapies such as ganciclovir and cidofovir, unfortunately, are limited by their toxicity profile. For example, ganciclovir can cause marrow suppression and cidofovir can cause renal toxicity. So alternative approaches to treat these viral infections are very important, and virus-specific T cells have already been proven to be safe and effective in treating these post-transplant viral infections, including CMV, BKV, adenovirus. There are multiple clinical trials at our institution and other institutions who have proven the safety of this approach and the efficacy, showing like response rates in the 70 to 80%. With no significant adverse events and no graft versus host disease, which is very important in the clinical setting. So at our institution, we generated these biobanks of virus-specific T cells, starting from patients who have recovered from certain viral infections, let's say who are seropositive for BKV. We would isolate the PBMCs, co-culture them with viral PEP mixes and cytokines to generate these virus-specific T cells, and then freeze and generate these biobanks of VSCs to be used when the patients need them. And we have published our experience with BKV-specific T cells treating JC virus-related progressive multifocal leukoencephalopathy, and more recently also published our experience with BKV-CTLs treating BKV cystitis. Again, in both settings, both for PML and BKV cystitis, these BKV CTLs have proven to be very safe and very effective with response rates in the 80% range. So during the pandemic, we started thinking also, why don't we apply this technology to treat COVID-19? So, following the same approach, we developed a methodology to expand virus-specific T cells from recovered donors. So, we start with a patient who has recovered from COVID-19 infection, isolate the Buffy-coat, the peripheral blood mononuclear cells, and then co-culture them with viral pep mixes that span the spike protein, membrane protein, and nucleocapsid protein of the COVID-19 virus. And then also with cytokine cocktails such as IL-2, 4, and 7, and then generate these virus-specific T cells that are specific killers of COVID-19 infected cells, expand them further, and then freeze and generate these biobanks again to treat patients with COVID-19. So after proving the success of this approach in the preclinical setting, we initiated a clinical trial at MD Anderson. It's a phase 1 clinical trial testing this approach in patients with mild COVID-19. So far, 15 patients were treated to date. We have proven the safety of the approach, and we have seen efficacy. All the patients who have been treated on this trial have been discharged from the hospital in good condition, and we have not seen any evidence of cytokine release syndrome or graft versus host disease or any other adverse events. However, despite the success of virus-specific T cells, there remains a very common clinical problem that we encounter in the clinic, and that's the use of corticosteroids. Corticosteroids, as we know, are very commonly used in the post-transplant setting to treat multiple complications such as graft versus host disease. And unfortunately, the use of corticosteroids can cause further immunosuppression and lead to increased risk of viral infections. Steroids also, as we know, are being used for the treatment of COVID-19 infection, especially in moderate to severe disease, in order to prevent lung inflammation and scarring. So the problem is, while these patients are being treated on steroids, we cannot enroll them on the clinical trials testing the safety and efficacy of virus-specific T cells. Why? It's because steroids are usually lymphocytotoxic, therefore they can cause apoptosis and killing of virus-specific T cells. So usually these patients are immediately excluded from the clinical trials if they are on steroid therapy. So a possible solution we thought of is to use CRISPR technology to delete the gene that encodes for the glucocorticoid receptor in virus-specific T cells. So the gene that encodes for the glucocorticoid receptor is called NR3C1, and we wanted to use CRISPR to knock it out. And we asked ourselves 2 questions. By doing this, do we really render these virus-specific T cells resistant to steroids? And do we affect their function or phenotype by doing so? So to answer the first question, we designed 2 different guide RNAs to target NR3C1. And we obtained a very efficient knockout of the glucocorticoid receptor, as you can see at the DNA level by PCR in the A panel, and on the protein level by Western blot in the B panel. You can see a complete loss of the protein. Functionally speaking, these GR knockout virus-specific T cells are completely resistant to steroid, as you can see in the C panel. If you focus on the upper panel here, you can see that the control virus-specific T cells, after treatment with dexamethasone, most of them become either apoptotic or dead, as you can see by annexin 5 staining and live death staining. So the steroids are killing the virus-specific T cells. But when we knock out the NR3C1 gene to make these virus-specific T cells resistant to steroids, indeed, after treatment with dexamethasone, you can see that most of them, all of them, retain their very good viability and do not undergo apoptosis. So this was very encouraging. This is further quantified in the D panel. You can see the percentage of live cells and the control virus-specific T cells after addition of dexamethasone, dramatic drop in viability. However, the GR knockout virus-specific T cells in green and purple after the addition of steroids, you can see that they maintain their viability very nicely. Do we affect their phenotype or their function by doing the GR knockout? And we don't. After GR knockout, we maintain the phenotype of these virus-specific T cells very nicely. The distribution of CD4 to CD8 is very similar after the knockout, and the expression of markers such as CD62L and CD45RA is also very similar following the knockout. The ability of these cells to produce cytokines such as interferon gamma, TNF alpha, and IL-2 is also not affected after the knockout. Even after addition of the steroids, you can see that they maintain their ability to produce these cytokines after exposure to the peptides. So we really were encouraged by these preclinical results, and we wanted to scale up this approach to take it to the clinic. So we used a 4D nucleofactor, a Lonza nucleofactor, and we scaled up this approach, starting from 3 million cells, going up to 25 million and up to 100 million cells. And we saw that the knockout efficiency remains very good, even at the 100 million dose level. You can see up to more than 95% protein loss, which was, again, very encouraging. And the second important thing you need to look at for a clinical product is its ability to expand ex vivo. And we were initially, wanted to know if by knocking out the the glucocorticoid receptor, do we affect the ability of these cells to expand? And luckily we don't. So you can see if you compare the solid lines to the dotted lines being the GR knockout virus-specific T cells, and the solid lines representing the wild type or Cas9 control virus-specific T cells, you can see that their ability to expand is very similar at the 3 dose level, 3 million in green, 25 million in blue, and 100 million in red. So again, this tells us that this approach can be scaled up in our GMP facility and can be taken to the clinic. In order to manufacture a clinical product, you need GMP grade materials. So we would use the high fidelity Cas9 called SPiFi, which is GMP grade provided by Aldevron, and a GMP grade guide RNA from Synthego to target the glucocorticoid receptor gene. So using these materials and using the scale up in our GMP facility, we're already preparing for this next generation clinical trial testing GR knockout SARS-CoV-2 specific T cells. So again, following the same schema, starting from a recovered donor who has recovered from COVID-19 infection, getting the buffy coats, the peripheral blood mononuclear cells, co-culturing them with the viral pep mixes and the cytokine cocktail to generate virus-specific T cells against COVID-19, and then introducing this large-scale knockout of the glucocorticoid receptor, and then expanding these cells further before we can freeze and biobank them in our GMP facility to be able to produce these off-the-shelf virus-specific T cell products that are resistant to steroids. And we are planning to use these cells in patients with moderate to severe COVID-19 infection, these are the patients usually who are treated with steroids to prevent lung inflammation and scarring. So we're very excited about that. The trial has been IRB approved and is being reviewed by the FDA. And another clinical trial also testing GR knockout virus-specific T cells to treat stem cell transplant-related infections such as CMV, BKV, and adenovirus is also underway. So moving on to the second part of this talk, focusing on CRISPR-modified CAR-NK cells. As you know, at MD Anderson, we have a platform to engineer natural killer cells or NK cells from umbilical cord blood. So we obtain umbilical cord bloods, we isolate the cord blood derived NK cells from them, and then we expand them with cytokines and feeder cells, and then transduce them with a retroviral vector to express a chimeric antigen receptor on their surface, and also produce cytokines such as IL-15 to enhance their persistence and proliferation. This was the first in-human clinical trial led by Dr. Rizvani at MD Anderson. And you can see that in this particular clinical trial, this was the CD19 CAR product. We expand the cells after the CAR transduction, and then the patients in the meantime would be receiving lymphodepleting chemotherapy with cyclophosphamide and fludarabine, and then on day 0 would receive the cell infusion. This was a very successful phase 1 clinical trial where we saw 8 out of the 11 first patients enrolled on the phase 1 arm achieve a complete remission. These were very heavily pretreated patients and with a difficult diagnosis such as Richter's transformation, who have failed multiple previous lines. And in multiple patients, we saw complete remissions. This is the example of patient number 5, who prior to admission, you can see the PET scan lighting up like a Christmas tree. And at 30 days post-CAR and K-cell infusion, you can see complete resolution of these PET-avid lesions. So despite the success of this program, we are starting to see relapses after CAR and K-cell infusions, very similar to what we see in the CAR T-cell field. So these cells are effective, but eventually some of the patients relapse. So we want to see if we can enhance the function of our CAR and K-cells even further by targeting checkpoint molecules. And the first checkpoint molecule we went after was cytokine-inducible SH2 containing protein, or CIS. This protein is encoded by the CIS gene, and it's a negative regulator of JAK-STAT signaling in both T cells and NK cells. And it's been shown very elegantly by Nick Huntington group in Australia that knocking out CIS in murine NK cells can release the brake on their activity against tumor cells. So we wanted to do the same, but with human primary NK cells that have been engineered with chimeric antigen receptor. And we were able to show that this approach is very effective in vitro, but also in vivo in a Raji lymphoma mouse model. We saw that these CIS knockout CAR NK cells can improve tumor control and survival in the mice. And this is a non-curative mouse model, so usually the mice really don't survive beyond a couple of months. But some of the mice that received the CIS knockout CAR infusion were able to survive more than a year. And when we sacrificed them later, we saw no evidence of lymphoma in their organs, as opposed to mice that received the wild-type CAR NK cells or mice that received the Raji lymphoma alone. So taking this approach hopefully to the clinic, we're preparing a clinical trial testing these next generation CAR NK cells for cancer immunotherapy, starting with cord blood, isolating the NK cells, CAR transducing them, and then expanding them, and then introducing this large-scale CRISPR knockout to delete some of the negative regulators of NK cells, such as CIS, TGF-beta receptor 2, and other negative regulators that we're going after, expanding these gene-modified CAR NK cells and then freezing them for use, for off-the-shelf use when the patient need them. So in conclusion, CRISPR-Cas9 technology is a very powerful technology that can be applied to enhance the function and activity of adoptive cellular therapies. And we have proven the efficacy of this approach in both virus-specific T cells and CAR NK cells. And we're very excited for the next generation clinical trials that are in preparation. So with that, I would like to conclude and thank everyone who has been involved in this work as delineated on this slide, and then especially the patients who have been involved in the clinical trial. Thank you very much for your attention, and I'd be happy to take questions if there's time. If not, then I can answer the chat. Thank you.
Yeah.